All right. Well, hey, everyone. Welcome to the second segment of TCSF's Family and Researcher Roundtable. My name is Casey Craig. Many of you know me as Stella's mom, but for those of you that are new to our community, I am also the Executive Director for the Cute Syndrome Foundation. I am really excited for today as we are going to learn more about an amazing research project that TCSF has had pretty close connections to since the very beginning as data was collected from the citizen efforts. If you may remember, a few years ago, our industry partner, Praxis Precision Medicines, used the SCN8A and 8A citizen data collected to help guide them when developing their Prax 562 clinical trial. And I find it just incredible that this data is utilized once again for this project. But I'm getting a little bit ahead of myself. So before I introduce our amazing guests, I'm going to go over a few housekeeping items. So this is a 3-part series. So this is 2 of 3. So make sure that everybody clears their calendars for next Wednesday from 12 to 1 Eastern Standard Time for our final segment. Today will be recorded. So if you feel like this is kind of like a water hose of information coming at you, you can always go back. We'll have this housed on our website so you can go back and listen at any time. Also, talking to Jan and Jillian, we really want this to be an interactive discussion. So we would encourage everyone to ask questions. You can type your questions in the chat box, and I will keep an eye on the thread throughout the presentation today. Also, due to the screen size and background noise, it's best for all viewers to keep your camera off and to stay muted. I also want to take this time to thank our platinum sponsor, Neurocrine Biosciences, for sponsoring this series. Okay, so let's meet our presenters today. I am thrilled to welcome researchers Dr. Jillian McKee and Jan Magielski of the Children's Hospital of Philadelphia. Today, they will present their latest SCN8A project, where they leverage both a database of 361 new and previously published individuals, as well as longitudinal citizen data on 79 individuals to characterize genotype-phenotype correlations, seizure trajectories, and anti-seizure medication effectiveness. All right, so Dr. McKee and Jan, the floor is all yours.
Great. Thank you so much, Casey, for having us here today and for that very kind introduction. As Casey mentioned, this is kind of works in progress. So we would love for this to be interactive. If anyone has any questions, any comments, any feedback, please feel free to put it in the chat, shout it out. We would love to hear from you to help us guide our next steps, as this is still very much our active ongoing research right now. Okay, let's get this slides ahead. There we go. All right, so just to start, I'm going to review some background about SCN8A, which is probably not needed for this group of this audience, but just some very basic background, and then talk about how we think about characterizing the phenotypic spectrum. And I'm going to tell you how we are doing this in 2 different approaches: statically, by collapsing over time and looking at the general overall phenotypes of individuals with SCN8A, and then also longitudinally, looking at how individual symptoms change over time to kind of get a sense of the natural history of the disease, but looking retrospectively at electronic medical record data. Then we're going to talk about how we can reconstruct seizure histories, outlining how dynamic the seizures change over time in SCN8A, and then also assessing treatment responses. Can we come up with a comparative effectiveness platform, like where we can assess how different seizure medications are effective in SCN8A as a whole, and in gain versus loss of function, people with different specific variants. So just a broad overview of what we're going to talk about today. So to start, where are we now? As many of you know, SCN8A was first isolated in mice in 1995, but the role in the human brain was first described in 2012. So it hasn't actually been all that long in the grand scheme of medical history that we have known about this condition, but we already know a lot. We know SCN8A is a relatively common cause of early onset developmental and epileptic encephalopathies, and it makes up about 2% of all DEEs. And it's associated with a very broad spectrum of disorders, including focal epilepsies, early onset epileptic encephalopathies, generalized epilepsies, movement disorders, and even isolated intellectual disability or autism. So what we're trying to understand is we're trying to use phenotypic science and data integration from multiple different sources to better understand these disease histories over time. We want to identify the unique features of SCN8A compared to other pediatric epilepsies and other genetic pediatric epilepsies. We want to explore genotype and phenotype correlations, so loss of function versus gain of function variants, and then also look at some of the commonly recurrent variants, so variants that come up again and again in the population. And then we want to assess medication response and try to get a sense of the comparative effectiveness of different treatment strategies. So a little bit about our approach. We're combining data from collaborators at the Danish Epilepsy Center, Citizen, and then our local medical center at CHOP, where we're followed a small but very well phenotyped group of SCN8A patients. Then we assign HPO terms, and I'll get a little bit more into what HPO means for all individuals in all different time intervals for which we have data. And then we perform computational phenotyping, reconstruct their seizure histories, and then get a sense of the comparative effectiveness of medications. So my research mentor, Ingo Helbig, loves to display this slide towards the beginning of some of his talks, just to convey the immensity of data we're able to access through electronic medical records. So this plot shows over a million neurology notes across over 100,000 individuals, capturing over 120,000 patient years. So the scale of this data is truly incredible, but it requires very rigorous methods for transforming and analyzing the data. And this is where data harmonization and different ontologies come in. So there's different approaches to phenotyping. And I'm just going to say right off the bat that none of them are perfect. There's always a trade-off between sample size and data granularity. So we obviously want to maximize both, but that's impossible when you get into these impossible approaches up in the top right. And then methods that have low sample size and low data granularity are just not useful. So those are irrelevant approaches. So then we're left with approaches that fall along this diagonal line. So large-scale phenotyping, as in this neighborhood analysis, uses very, very large sample sizes, but then lacks the granular detail to really get a sense of how things change over time. And then in contrast, natural history studies record data, super detailed data about every individual over time, very, very intense studies. But they can only, because of the level of detail required, they can only handle pretty small sample sizes. So as this is a constant trade-off, which method is best? So I'm hoping to convince you that by using real-world data that spans the spectrum, we can aim to get the best of both worlds. So I'm going to pivot and look at some of our different data sources. So we're going to talk about data from three different sources today that kind of span these options. First, we have access to large-scale static phenotyping with data that was shared to us through our collaboration with the Danish Epilepsy Center. It's able to capture genetic and phenotype data across a large number of patients, but it's at a single time point. So it doesn't tell us how the symptoms change over time. So we're calling this our static cohort. In the middle, there's Citizen, which consists of curated medical record data with moderately large sample sizes and level of detail. And then finally, there's our internal CHOP cohort, where we have access to very detailed medical records, but our numbers are pretty small. And together with the Citizen data and our data, we're calling this our longitudinal cohort. And I just want to take a moment to comment on registry data as well. As many of you know, there's a large SCN8A registry run by Dr. Mike Hammer. And this data set is very rich as well. It contains caregiver reported information on a large number of individuals. And we strongly believe that this data set is complementary to ours. We think they'll both lead to important results, and that caregiver reported information often captures different aspects of a condition than our electronic medical record data that we're using primarily. And with this electronic medical record data, it's already there. It's being recorded through routine care. So we should use all the data we have access to help understand these genetic conditions. So studying this so-called real-world data that can accrue over time has led to important discoveries in other conditions, and think it will do so for SCN8A as well. So just a little more detail about our data sources. We've been collaborating with the Danish Epilepsy Center on a static cohort of individuals with SCN8A, and we'll present some of the preliminary findings today. This large-scale phenotyping effort has been led by Elena Gardella and Reike Moeller, with much of the phenotyping and curating being done by David, Francesca, and Roberto. As most of you know, Citizen is a company now owned by Invitae that aggregates and curates data from medical records and provides it to researchers, physicians, and pharmaceutical companies with the goal of advancing targeted therapeutics for rare disease. And here I'm just showing you some of their partners, including, of course, the Cute Syndrome Foundation. And just want to share off the bat that this has been a bit of a whirlwind analysis for us. We actually only received the complete Citizen data set about 2 weeks ago. So we're going to be showing you our preliminary findings from that. But just know that there's much, much more to come, and we'll have more to share with you even by AES this year. And then finally, we follow a small cohort of SCN8A patients at CHOP through our Epilepsy Neurogenetics Initiative. And on this cohort, we have access to detailed clinical histories, medication prescriptions, lab work, imaging, and EEG results. So before handing this talk over to Jan to review some of our preliminary findings, I wanted to briefly describe computational phenotyping. In our work, we leveraged the HPO, or the Human Phenotype Ontology, to perform our clinical phenotyping. And this is a hierarchical structure of clinical terms, as you can see here with this pretty figure showing how all these terms are interrelated. Often in medical records, symptoms are documented at different levels of detail. So for example, for the same patient, one note may say absence seizure, but then another one may say generalized seizure. And people may not realize that they're actually describing the same thing. So in order to translate across that, you need some sort of hierarchical language dictionary to help you know how closely related different clinical terms are. So that's what the HPO lets us do. It has a branching logic that tells you an absence seizure is a type of generalized seizure, so that we know how all of these different terms interrelate, so that we can calculate how similar or different two different individuals are. So that's the benefit of the HPO. And all right, I'm going to hand the next portion of this talk over to Jan, who is one of our wonderful students who's really been working hard on SCN8A, and he's going to share some of his preliminary static analyses.
Yeah, thank you, Jill. So now we're going to start with our static cohort, like Jill said, and we're going to take a look at, the breakdown of the variants and the general characteristics of the population. So as you can see on the left, we were able to gain over 350 individuals for the static cohort, majority of whom are from the previously published papers, and the rest, like Jill said, were taken from the Danish Epilepsy Center group. In our cohort, we have 124 individuals with gain-of-function variants and 23 with loss-of-function variants, with the remainder being of unknown functional consequence. And as you can see, also the vast majority of individuals has de novo variants. On the right, you can see the phenotype frequency plot. And as expected, what immediately pops up is terms like seizure, neurodevelopmental abnormality, or some behavioral phenotypes that I will describe in more detail as we analyzed each specific subgroup of this static cohort. So now we're going to take a look at our variants in the cohort. So on the right, you can see the SCN8A protein stretched out, and each line represents one variant that we observed in our cohort. And the higher the line is, the more individuals have that given variant. So you can see that we identified quite a number of recurrent variants that we labeled here, with the most common ones being on position 1872, 1877, and 1475. Yes. Overall, we were able to harmonize over 10,000 clinical terms, which, that's the number of following the harmonization and propagation that Jill mentioned. So we had base terms that we then propagated, with more general terms that were present on that phenotypic tree that Jill showed for all individuals, and an average of 30 terms per individual was recorded for that static cohort. And this enabled us to observe differences and similarities across different subgroups in our cohort, which I will discuss now. First, genotype-phenotype correlation. So, one thing that's important to know in conditions like SCN8A is whether by knowing if someone has a gain of function versus loss of function variant has any, phenotypic consequences. And we see that it does. There are a few key differences that we can see here. So we can see that for gain of function variants, we have higher frequencies of focal seizure, bilateral tonic-clonic seizure, and motor seizure in comparison to loss of function, for example. But we can see that, the frequencies of intellectual disability or delayed speech language development are of similar level, if not higher, for loss of function variants. And now we can take a look at more specific analysis of that. So to orient you with that plot, on y-axis, we see the frequency of phenotypes in gain of function variants, and on the x-axis, we see frequency of variants that are, for everyone else in the cohort. So this phenogram also displays, which values are actually significant association. So all red dots are significant, and the bigger the dot, the stronger the association. So we can see that in addition to the seizure types I mentioned previously, so bilateral tonic-clonic seizure and focal onset seizure, we also see some movement disorder phenotypes, including incoordination or dyskinesia and hyperkinetic movements. We did the same analysis for loss of function, which you can see that we have a bit, smaller pool of individuals than gain of function. And again, the frequency for loss of function is on the y-axis and for everyone else on the x-axis. And we can see some differences, right? We see that there's absence seizure and generalized onset seizure that is more prevalent for loss of function variants, while we also observe more of a behavioral phenotype. We see aggression or impairment in personality functioning that was also significantly more likely to occur in people with loss of function variants in SCN8A. Then we performed the same phenogram analysis, but for those two of the most common recurrent variants that I mentioned on the first slide. So first, looking at variants at position 1872, we were able to observe that, apart from the gain of function features that I mentioned, because it's a gain of function variant, so we would expect that, bilateral tonic-clonic seizures or focal seizures would pop up as significant here. We also see that status epilepticus is a significant feature associated with that particular variant. In addition, we see some, again, movement disorder phenotypes like hypertonia or upper motor neuron dysfunction that were more likely to occur in those individuals carrying this variant. Then we performed the same analysis for the second most common recurrent variant. And it's interesting to compare with the previous one because here we also are dealing with a gain of function variant, but we clearly see different features. So apart from bilateral tonic-clonic seizure, which is a common theme for gain of function variants, we see that there's also a stronger behavioral phenotype with aggression present or or incoordination that we also saw previously. So we can, by analyzing those recurrent variants, we can appreciate the diversity of symptoms even within one category of functional consequence of a given variant. Here we see stronger behavioral phenotype, while the previous one was, more neurodevelopmental or motor symptoms related phenotypes.
Yeah, and I'm going to interrupt you for just a second. We have a question in the chat box.
Yes.
The question is, with the reoccurrent variants, are you able to predict outcome or progression of a patient's condition?
So we haven't quite looked at how seizure progress in terms of each specific variant. Jill will talk more about what we know for now for SCN8A as a whole, but definitely we will, as we progress with analysis, we'll look more into how, if there are any differences for specific most recurrent variants or, gain of function versus loss of function. But yeah, we aren't there yet, but it's a good thought for sure.
Sounds good. Thank you.
And yeah, lastly, for phenograms, we looked at a comparison with SCN2A, a different condition that's also affecting sodium channel, but just a different one. So before I jump into the results, on this plot, whatever's on the right from the dashed line in the middle is more likely to, and red is more likely to occur in SCN8A, and whatever's on the left, more likely to occur in SCN2A. So we observed that features that are more common in SCN8A were focal seizures, bilateral tonic-clonic seizures, and motor seizures, some movement phenotypes like tremor or muscle weakness, as well as visual impairment and speech and language development. So you can see that the dot on the very top is delayed speech and language development, which was the most significant association here in comparison to SCN2A. On the other hand, we see for SCN2A that we have more generalized onset seizures and focal tonic seizures, as well as more autistic behavior. So that's one of the examples of what we can do when we look at across conditions. It may be useful whether there are any signatures of one condition versus the other. Now, lastly, to close the portion of our static analysis, we began to look at clinical subgroups, and we started with developmental epileptic encephalopathy, which is DEE. And here, which probably doesn't come as a surprise, all of the features are very frequent because it's, given the symptom severity of DEE, it makes sense that seizures and neurodevelopmental abnormality and related terms would be high in frequency, but we're hoping to look more into other clinical subgroups for the future as well.
Thank you. Sorry to interrupt. I was just going to throw in something. The last slide that you had up, the comparison between 2A and 8A. I find this interesting, especially as we start looking at, our clinical trials and having things that are approved treatment for SCN8A. It'd be interesting if something like this could be very powerful for the FDA if we were comparing maybe a genetic mutation that already has FDA approved treatments versus SCN8A. And there might be a lot of red dots on SCN8A and here we are with no treatment at this point. So yeah, that's just something I was thinking of as we're kind of going through this. This might kind of be able to see on an actual graphic the severity of SCN8A. We also have a couple of questions really quick in the chat before we switch over to Jillian. So the first one is, with recurrent variants, do you know if things like aggression are due to the variant or possibly due to them being on the same medication, such as like side effects?
So that's a good question. Probably we don't, in the sense that it's hard. So with medical record data, it's difficult to tell whether something is recorded, say aggression, right? And we can also see like when someone was on a given medication, but whether it is a cause or not, like it's impossible for us to tell at this point. And probably it is more granular data sets that Jill was saying with like natural history studies. Maybe that's where we could actually pick up on that, but not with what we have as of now. I don't know, Jill, if you have any other.
Yeah, that's a really great and a really tough question. And you're kind of getting at one of the problems we encounter when you talk about a natural history of a condition. What even is the natural history? We don't let children live in a vacuum, and we don't not treat them to see what happens over time. So all of our natural history is really not entirely natural, because we're always throwing medications at children and trying to treat them to the best of our current knowledge. So we don't really have a sense necessarily of how like seizures or other symptoms change over time without intervention, because we always intervene. We're not going to not try to help someone. So yeah, it's a great point, and we just don't know at this point with large enough data, and having timestamp data where you can see how symptoms relate to when certain treatments were started and stopped. You get a better sense of it, but none of this data is pure because we're studying real children who need therapies.
Yeah, that's a good point. We also have another question. Are your results and features of gain of function versus loss of function identified different or similar to what has been published by the Danish group, the Hammer Registry, and the core treatment guidelines data?
So we were using to answer the first part of the Danish group. So we are collaborating with them on this. So the data they used, what we're using here is the updated version of what they used to publish in the paper. So it's an expansion of that, but we haven't really seen any major differences that I would know of. The other two, I'm not sure, Jill, if you
Yeah. So, I guess the broad answer is overall similar. But we're still at early stages of really delving into a lot of this. So there's definitely the potential when we look at, there's like a lot of red dots on a lot of these plots, if you notice, and we haven't totally scoured every single one of them to see whether it's a novel or previously reported. The ones that pop out the most are the commonly associated symptoms with gain and loss of function, but there's probably some other things in here that may be new and interesting too.
And the things that Jan just went over is just the static information from the Danish group as well. And so now we're going to kind of move into a little bit of talking about, the longitudinal data, which would be more kind of in the next, those two other things that you were talking about. And, I know right now you're definitely in early stages. We might have some more information before our gathering in December, but this is definitely in general, like, when did you guys start this project again?
We started the SCN8A in the spring, but we only got the longitudinal data a few weeks ago. So
yeah. So you've done a lot so far just in the short amount of time that you guys have worked on it. But okay, good. Well, I'll quit interrupting for now and let you move on.
Oh, that's great. Yeah. So now we're gonna, like Casey said, we're gonna move to longitudinal analysis. So based on the data that we got from Citizen, after propagation, we collected overall of over 160,000 phenotypic terms in 79 individuals across a total of 569 patient years. On the left, you can see the plot displaying all of that data. So it's the same plot that Jill showed at the beginning, but just for our Citizen cohort. So each row of dots is a different individual, and then each dot is representing a clinical record, a time-stamped clinical record for each individual. So the longer the line, the more we have on a given person, and the shorter the less we have. Now we're gonna move on to longitudinal phenotypes. So before I actually describe what the plots are showing, let's describe how the plots are set up. So on the y-axis, we have number of individuals, and on the x-axis, we have age in months. And red color shows that phenotype is present, and blue that phenotype is absent. And you can see that all of those plots have the same shape. And the reason for that is for each month, we have a given amount of data, and then what's just changing is the proportion of present versus absent traits. So you will see that the shape of the overall graph stays the same, but only the proportion of red and blue will shift. An additional thing to mention is that, which is always the case with data like this, is we have higher amount of data for the earlier stages of life with lower amount of data for later stages in life. Now to describe actual results. So for seizure, neurodevelopmental delay, and hypotonia, you can see that it's pretty much present at all stages of life for individuals with SCN8A. with neurodevelopmental delay and seizure being, very, very, very prominent feature throughout life. Now when we shift to another group of phenotypes on the next slide, we see that features like abnormality of the digestive system or respiratory system or sleep abnormality, which is frequent, these are features that, families frequently report in these conditions. We can see that there is some age dependence there with, those features being more frequent earlier in life, especially for respiratory system abnormality and sleep abnormality. But overall, it still remains, present across all of the age groups that we were able to look at this point. So now, if there's any interest in particular traits or those clinical traits and how they progress over time, you can let us know and we can take a look. But now we will discuss seizure landscape of SCN8A, and I will turn things over back to Jill.
Great, thanks, Jan. So now moving on to seizure characterization. One important question people often ask us is, when do seizures start? And like, if you have a child at a given age and they haven't seized, like, what's the likelihood that they're going to go on to have seizures? So here what we're doing, and this is still using the static Danish cohort, we're plotting the cumulative proportion of individuals who have seizures across time, and showing this for the overall population in blue, broken down for those with gain and loss of function variants as well, with gain of function in green and loss of function in red. And you can see here that individuals with gain of function variants tend to have seizures earlier in life than those with loss of function. But looking at the seizure onset in any given individual, it's a little tricky to tell. These differences become apparent when you look across the population of individuals. So that was from the static seizure onset data from the Danish group. With the longitudinal data from Citizen, which I'm showing you here, we can map seizure severities over time for all individuals. So here's similar setup to the prior plot Jan was showing you with the presence or absence of data. But here again, each row is an individual and each dot is a recorded seizure frequency in the medical record. And you can see that the density of the data varies for different patients. Some people only have like a couple data points and some have many data points. And most have at least more than one data point. And that you can see that the severities vary across the lifespan in different individuals. Our data obviously gets sparser as people get older. We don't have as much frequent seizure data on people in like their 20s. But you can see that there's no clear pattern here with seizure frequencies like spiking at a certain age when you look at the data this way, at least. But we plan to look at this in more detail for people with gain versus loss of function variants, for variants with different functional consequences, and then for the recurrent variants as well. So then now looking at even more detail at our local CHOP cohort, we can reconstruct seizure frequencies in monthly time bins and map out how seizures change across the lifespan. So here, each bar I'm showing you is 1 month from birth to age 3 years, showing the proportion of individuals with different seizure frequencies. The connecting lines show how individuals' seizures change between months, with blue connecting lines meaning an individual's seizure frequency improved, and yellow means that it worsened from 1 month to the next month. So using this approach, we can compare how seizure trajectories are different across different genetic epilepsies. So for example, here I'm showing you SCN8A compared to STX-BP1 and then SCN2A. And we found this has been a helpful approach for not just like having a static like seizure onset is at this age, but really getting a granular idea of how seizure frequencies change over time, which can be helpful both for counseling families, but also for clinical trial readiness. We really need to have a sense of when are the best times that we could potentially intervene with a novel therapy to show the best response in a clinical trial. So next we can also extract from the Citizen data set time-stamped medication data. So here I'm showing you from Citizen, and we're plotting the frequency with which different medications were prescribed over time. And you can see there's a lot of them here in the citizen data set. And we've also done this for our local CHOP cohort as well for SCN8A, but also some other conditions. And these are just small number of pilot data, which we're working on expanding. But here I'm showing you SCN8A as well as 2 other sodium channel related epilepsies. And you can see how the treatment strategies differ. For SCN1A, a lot of these children have Dravet. You can see that there is no oxcarbazepine, for example, which is trileptal, which is a sodium channel blocker, because that would not help these children. Whereas for SCN8A, you'd see a lot of sodium channel blockers, which are often very effective in children with gain of function variants. So it's a nice way to get a sense of how different treatment strategies change. And then we can also combine that, the medication data, with the actual seizure frequency data in a particular individual and then across the population to get a sense of the comparative effectiveness of different medications. So what I'm showing you here on the left is a schematic of a single individual's seizure frequency over time on the top, and then the different medications they were prescribed with start and end points on the bottom. And what we can do is look at particular time periods where either seizure frequency was reduced, which is highlighted in green on the plot, or periods where seizure freedom was maintained, which is the period in blue, and then compare which medications the children were on during those periods to all the other time periods when they were on different medications. And we can calculate odds ratios of the effectiveness of different medications. So we've done that in our local CHOP cohort in a small group of only 15 children with SCN8A. And we're going to try to do this for Citizen as well for the time bins that we have this data. And we've done it for some other conditions as well. So here I'm just showing you on the left SCN8A compared to STX-BP1, just as an example. And you can see for SCN8A, as we'd expect, oxcarbazepine or trileptal, a sodium channel blocker, is much more effective at reducing seizure frequency than it is in STX-BP1, for example. So this is a nice way to get a sense of which medications might be most likely to be effective in different genetic epilepsies. And then once you have a large enough population, you can actually do this in different subgroups too. So in those with gain of function, loss of function, specific recurrent variants too, and then looking at different biophysical properties too.
Cool. Jillian, I do have a question from the audience. So it says, is the data used on what a doctor will write in a clinical statement? She said that they've looked over some of their stays regarding the citizen data. Some of the things that they write in the notes are different than what I believe to be happening. So what a doctor may write may not be agreeable between a parent sometimes. So in general, what she's asking is, are you guys using like the MRI and EEG reports in addition to the clinical notes, or how does that work?
So that's a great question and a great point. So this data isn't always perfect. So the citizen data, when we receive it, is extracted timestamped clinical terms, which are extracted from the notes. So we don't actually, for privacy reasons, we don't actually get like the clinical notes themselves to know exactly what was said, but it's extracted and coded as a clinical symptom and a certain time. We can compare that to, we also do get some of the terms from the EEG reports and the MRI reports, but this is just using those extracted clinical features. So this would be more heavily weighted towards the physician's perspective on what's going on rather than the caregivers, which is why having registry data as well to compare to is helpful, because we know those aren't always the same. Physician notes aren't perfect, and they may have a different caregivers probably have a different view sometimes on how things are going. So I think it is valuable to look at both of those data sets. But yes, to answer your question more specifically, this is just looking at what Citizen has coded as that feature at that time point, and that is coming from the notes. They record the EEG results as a separate, like a procedures file that we also look at, but not for these analyses.
Okay. Thank you.
Sure. So finally, I had briefly mentioned earlier that we want to look at how things like the phenotypes and the seizure frequencies and the medication response differ for individuals with variants that have specific functional consequences, so different biophysical properties of those channels. And to do this, we are leveraging Phoenix, which is the functional epilepsy nomenclature for ion channels. And it's a controlled dictionary mapping functional characteristics of ion channels and binning them into various levels of severity. So our goal with this is to combine comparative effectiveness with this functional data. It's because you can presume medications act on these channels in different ways, and it's logical to assume that different medications would affect different channels differently if those channels have different biophysical properties. So that's what we want to assess next. So here I'm just showing you seizure onset data broken down by functional consequence of the ion channel. And we're just showing you a few here, and this is still very preliminary. And those differences aren't that striking. We're planning to explore more functional effects and how they impact seizure frequency, severity, and medication response, as well as other symptoms too. So here is just an example of the comparative effectiveness in small pilot data using a very small population, showing that this sort of approach may be useful, at least for medication selection. So here I'm showing you two different medications, lacosamide, which is Vimpat, and levetiracetam, which is Keppra, and showing you the odds ratio. So those same comparative effectiveness odds ratios I was showing before for people with variants that have 4 different functional consequences. So like the variant changes the persistent current through the channel, or I know these all have like very complicated, very technical names, but the overall way to think of it is they change how that channel is functioning, either letting in more current, keeping it open longer. And based on the way these medications act, they have different effects. They work different on these channels that have these different properties. And at least with this preliminary analysis, it looks like it's true that, like, for example, Vimpat, lacosamide, doesn't seem to work as well for people who have the B type of channel, whereas it works better for the other ones. And Keppra also shows some differences. So this is very preliminary, but we do expect to see findings like this if you look across the whole population, because this is how we intuitively expect medications to work, right? They affect how the channel opens, either how long it's open, how fast it opens, how quickly it can reopen once it's closed. So this is exciting to us, because then if you know an individual has a specific variant with a specific functional consequence, it could potentially help you guide which medication to choose. So you don't have to go through that very painful trial and error of trying so many medications, which I'm sure many of you on the call have been through.
Yeah, probably all of us.
Yeah, probably.
Back up just really quickly. When we were talking about the different types of medications, there was a question that popped up. For those patients, it was that, I think it was maybe a couple slides before that. Is this after diagnosis, the drug tracking? This one. Yep.
So for most of these patients, it would be for after diagnosis, because that's when we would have the data. Although this is from our local CHOP cohort, so we follow some of these patients. So it may have been from before they were diagnosed as well. We have seen, like, for example, for like kids with Dravet syndrome, that before they're diagnosed, they are on trileptal and like sodium channel blockers that maybe they shouldn't be. And then as soon as the diagnosis is made, that medication is stopped. So for some of these patients, we would have data before the diagnosis, others we wouldn't. So it's mixed.
Okay, thank you.
And that's pretty much what we wanted to share with you guys today. I hope we've given you a little sense of like how we're approaching this, both statically with the Danish collaboration and all that very large scale, but less detailed data set. And then the citizen data, which is much more granular, but a smaller population. And then our very, very small population at CHOP, where we're able to very deeply phenotype these individuals. Would love to take any questions, but also just want to thank both our internal CHOP SCN8A team, which is Jan, me, Dr. Ingo Helbig, who you guys are probably all familiar with, our wonderful sodium channel oriented genetic counselor, Stacy Cohen, and then Julie and Michael, who are some data scientists who have helped us with this project at various points. And then also want to thank all of our collaborators, Citizen for the wonderful data, and then of course, Cure Epilepsy and the Cute Syndrome Foundation for helping support this work.
Yes, absolutely. So I'm looking at the chat. Let's see if anything pops up here. Thank you so much. And I'm really, really interested in a couple of slides before that when you were looking at like the different tracks, if you will. And gosh, how nice would that be if we were able to, you know, between this data and other data, like be able to say, okay, you're track A or track B, and let's move on to this drug and this drug, or don't do this drug. So yeah, I love this this graphic right here and what it shows us.
Yeah, I mean, that's the dream. If we could do that and be able to predict. Oh, let me see. I can stop sharing too, unless people are gonna wanna. Then I can see everyone else.
Exactly. There are a couple more questions in the chat box. I'm not sure if you're able to see those now or not, but so it says, is there any way to extract the cost of being an SCN8A patient from the medical records from Citizen, like the codes used for insurance?
Oh, that is a great question. Probably not from Citizen that I can think of, but we have worked for some other conditions with claims data sets, and then you can get a better sense of the cost of insurance. But for that, you need to have an ICD-10 code to be able to pull people with that specific condition. But no, we have not looked into the costs using the Citizen data set.
Yeah, and that's interesting. Madeline, that she was the one that asked that question. And I'm curious, I'm guessing maybe you're thinking if we have a general like cost of if you were to invest in this, maybe there's like another new and upcoming treatment that costs a lot of money. But if we were able to say, well, typically an 8A, you know, patient cost, I don't know, $1 million a year. And if you were able to spend this money on this other therapy that may be a little bit more risky, but, you know, could the benefits outweigh the risks? And then, yeah, Tammy had just kind of commented, I can't even imagine the cost of like, you know, I see the EOBs that come across for, you know, a ICU stay or an airplane ride. So I can only imagine if we added that up in Stella's short 8 years, what that might cost. But yeah, I agree.
Yeah, I'm sure it's astronomical. And that's when it's an important question, because when we're thinking about these more targeted therapies, they are very expensive, at least initially. So having some sort of cost analysis where we can show that the standard treatment is, well, it's split up into a bunch of hospital admissions, ED visits, clinic visits, medications, and each of those individually aren't that expensive. The overall care for one of these children is very, very expensive.
Yes. Yeah. Yeah. Madeline said that she's heard from many people from biotech that having that could be very valuable going forward. So I know that's not really, you know, what you guys are looking at right now in your information, but if there's a, you know, if you can lead me or, you know, our community into people that might be interested in that, we could definitely look at that too. Or if it's something that you guys stumble across, let us know for sure.
Yeah, definitely. Yeah, I think that's an important thing to do. And I've seen some other people talking about it, but I'm not, it's not our direct area. So it's not something I've looked into all that much, but I think it is something we should focus on a little more.
Yeah. Yeah. Very interesting. Any other questions? Yes, we have another one. Let's see here. Are you able to look at the variants that are more of a mix of loss of function and gain of function? So I think what she's referencing is if you have a particular individual that is not a proven loss of function, gain of function, but they're kind of finding themselves in the middle of these symptoms between these two phenotypes. Do you have, are you able to look at that and kind of give them some information?
Yeah, so we're hoping with Phoenix, which like looks at the different functional consequences of all these variants, we'll be able to have finer groups, not just gain of function, loss of function, but actually be able to tell what how the variant is actually changing the way the channel works. And then once we have enough people with specific variants, we can start to look at that and give you similar sort of information, but for variants that don't classically fit into one of those two buckets. And as Jan showed with the recurrent variants that he showed, even though both of them were classified as gain of function, they did actually have quite different phenotypes. So the gain of function, loss of function isn't like the be all and end all.
Yeah, very interesting.
Yeah, I think the only problem would be with the, just like Jill said, with the number of patients, probably that we would have to have enough to show anything meaningful on those, association or phenograms that I showed. Because otherwise, if it's like one or two people, it's very difficult to make any statements that are, true.
Yes, exactly. And I guess I can kind of put a plug in here too for those families that did the citizen outreach and enrolled their kiddos through that. Now would be a great time to kind of do a refresh for all of that information. So you have that too whenever you guys are looking at the longitudinal data for later on.
Yeah, definitely. I see Shelley's question there is how can caregivers help? And participating in the registries, whether it's My Camera's registry or Citizen or both ideally, is very helpful, because what we need to answer a lot of these questions is more data.
Yeah. And just to play kind of devil's advocate, what if your data and the things from the natural history data, what if they match up and say the same thing? Like what would that tell us?
What if citizen and natural history data say exactly the same thing? I mean, that would be really helpful, because you may not need then a super large scale natural history study before designing clinical trials. If like a smaller natural history study matches with the citizen, then you give you some hope that maybe you could rely more on the citizen data as a surrogate, like these, we call them like retrospective natural history studies, like just using electronic medical record data. It would almost be a validation of our approach. So that would be very helpful.
Also, we have a question from a parent saying, are you accepting 8A patients? Do they have to live near CHOP? So I think if they're saying if they wanted to come to CHOP to, either get a second opinion or get treatment, are your awesome doctors accepting? And
yeah, we see in our engines, or it's our epilepsy neurogenetics initiative. It's the acronym is ENGINE. In our ENGINE clinic, we see patients with all different neurogenetic conditions, mainly epilepsies, but other neurodevelopmental disorders as well. Many with 8A, and we would love to have more people come see us at CHOP, because then we can perform our more detailed, even if you enroll in our research projects, perform our more detailed reconstructions on more individuals. You do not need to live near CHOP to come see us. We have patients come see us from all over the country and from other countries sometimes too. If I can put like our ENGINE email in the chat, or Jan, if you are able to type in the chat, you can reach out and we can talk about scheduling. Like insurance stuff sometimes has to be worked out, but yes, we see people from all over.
That'd be great. Another question, have there been any documented cases of 8A variants being neither a gain of function or loss of function?
That is a good question, Jan.
I think mostly it's either that it can be mixed effects, or the functional consequence, it's unknown. So in order to be able to tell whether something is gain or loss or mixed, you have to perform, experiments on cells and record with various devices to be able to tell that, and just not all of them. It hasn't been done for all of possible variants, so there are, but many of them would be because they haven't been studied yet, or they have mixed effects.
The same parent said that their son is classified as neither per current research at MSU, and we don't really know what that means. So I'm guessing then maybe from what I'm gathering for your guidance is saying is that their particular 8A variant has not been proved to either be gain of function or loss of function. Would you say that's true? It just hasn't been researched in a lab?
That would be my assumption, but it's hard to say without like actually knowing what the variant is and like looking into it. But that would be my assumption, that it just not every variant has been studied in the lab yet.
Yeah, exactly. Okay. Any other questions? Yes, we have. Oh, yep, that's Jan putting in that email address there. So Megan, if you're interested, or anyone else, feel free to jot that down. And I can also share that whenever we're sharing our recording. I'll make sure that I have that posted as well. So that way families can get to that easily. Awesome. Well, we kind of did perfect on timing, by the way. Then, yep, Shelly shared our citizen efforts as well from our website, so we can share that as well too.
Great.
Yeah.
Right.
Thank you so much. I really, really appreciate you guys coming today, sharing what information you have so far. And then I know you guys have been crazy busy trying to get this ready to go for us. So I really appreciate it. And then I'm looking forward to hearing you guys speak again in December at the gathering, and hopefully have a little bit more longitudinal data from there. So really appreciate that. Oh, Shelly's saying, just as a kind of reminder, make sure you're going in and you're refreshing that citizen information, which I totally need to do that. I am guilty of needing to do that as well. So I need to put it on my to-do list. So thank you guys again so much. And thanks again to our platinum sponsor, Neurochem Biosciences. And then next week, families, so make sure you mark your calendars for Wednesday when we get to speak to Dr. Miriam Meisler. And she's going to talk about the differences between de novo inherited mutations, and then also kind of what that means for possibility of reoccurrence with future pregnancies, which I know that is a hot topic among families a lot. So I'm excited to get to chat with her. So, all right. Well, Jan and Dr. McKee, thank you so much for coming today. And we get to see you both, right? In December. Awesome. Super excited. All right. Well, thank you, everybody. We appreciate you being here, and we'll see you next week.
Great. Thank you so much for
you.
You're welcome. All right. Bye-bye.
Bye.